Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, Supercharge your Olink workflow with the right liquid handling tools. My name is Dora Wells, and I'll be moderating today's presentation. I'm delighted to be joined by today's speakers, Dr. Anne Hammerstein, Product Manager at SPT LabTech, and Dr. David Corney, Principal Scientist working within the Next Generation Sequencing Department at Azenta Life Sciences. After the presentations, we'll move on to our question and answer session. Please feel free to ask any questions for the Q&A session at any time during the webinar. You can submit your question at the speech bubble icon to the left of your screen. And now, without further delay, I'd like to hand over to our speakers for today's presentation, and I'd like to thank them for presenting to us today. Please go ahead.
Thank you for the introduction, and thank you for joining us today for today's webinar. My name is Dave Corney, and I'll be talking about biomarker discovery using Olink and miniaturized liquid handling. So first, we are going to talk about some of the benefits and challenges of protein biomarker detection, and then we're going to switch gear and talk about how we can actually perform protein biomarker detection using the Olink platform. So before I get into that, I just wanted to give a quick introduction to Azenta Life Sciences. You may have heard or may have worked with a number of these companies on the left-hand side here over the past, Brooks Life Sciences, GeneWiz, Fluidex, and so forth. And about a year ago, we rebranded to Azenta Life Sciences, which signals our commitment to helping customers across a wide range of applications. These applications include not only genomic services, which we will talk about in greater detail today, but also sample sourcing, sample storage and logistics, as well as a suite of consumables, informatics, and consultative solutions. So I mentioned that we have a wide range of services that we provide under the genomics umbrella. Today, we will talk about next generation sequencing and qPCR, but we do also offer other services such as oligo synthesis, gene synthesis, and so forth. But in terms of next-gen sequencing, we really do have a wide range of platforms in-house and a wide range of applications that we can support, whether that's RNA sequencing, whole genome sequencing, targeted sequencing, and so forth. Now, for today's talk, we will be talking predominantly about protein biomarker discovery, but at the end, I will come back to talk about how we can combine a wide range of these applications together for a multi-omic based approach to detect a wide range of analytes. So let's talk about some of the benefits and challenges of protein biomarker detection. And I wanted to start out really going back to the fundamentals and talk about the basics of biology. And of course, we know the central dogma of DNA, which is undergoing replication as well as transcription to RNA. The RNA is then being translated to protein. And of course, historically, we've always thought of protein as being the functional output of the genome. Of course, with transcripts such as microRNAs and other non-coding RNAs, we now understand that RNAs also have an important function. But I think is important to look at these three different biomolecules quite separately when we consider their relevance to disease and biology. So when we consider DNA as a very static indicator, we're looking at maybe variants in the germline that could impact predisposition to a disease, but is somewhat static. Of course, when you consider somatic variants, then there's a greater play of involvement of those somatic variants in cancers, for example, and that can be tracked and used as a prognostic during disease progression, but is pretty static, whereas RNA is pretty dynamic and protein the most dynamic of all. Now, when we take that understanding and also look at the complexity of these different molecules, we see that they also increase in complexity too. So the DNA is somewhat of a small consort of molecules, around 20,000 genes, increasing to 100,000 transcripts and many more than 100,000 proteoforms when we look at the proteins. So taken together, I think this is maybe one reason why we focused our attention up until now predominantly on DNA and RNA. So when we look at the field of how genomics has progressed, we see a number of parallels to proteomics. Uh, starting in a few decades ago with Sanger sequencing, with a very low throughput workflow, somewhat challenging interpretation, and the same can be said for proteomics, where mass spec was one of those very first technologies to look at proteins. And then we get into this phase of using microarrays, which have the advantage of much higher throughput and being able to look at many more samples, looking at many more proteins or many more RNAs, for example. But the challenge there is that they're only able to detect known genes or known proteins. And this is why the advent of high throughput sequencing really has expanded the ability to perform high-plex and high-throughput RNA and DNA sequencing. It's allowed us to identify novel genes and novel proteins and variants with high throughput. So as I look at these two fields together, clearly we're in this post-high throughput sequencing era for genomics. Whereas for proteomics, we're now just getting into this phase of being able to perform high throughput analysis of proteins. So when we consider some of the questions we might answer, and I'd like to answer within proteomics, typically we're interested initially with an association of a specific protein with a disease state. So this may start with trying to discover novel proteins and novel isoforms, but very quickly we want to translate that to how does that impact biology. Sometimes we may be interested in the abundance or the turnover of proteins and how this may impact the concentration and the function of that protein. We're also interested in where that protein may be expressed. So is it expressed cytoplasmically? Is it bound to a specific organelle within a cell? Is it expressed in a specific tissue? Are these proteins modified after translation, and how do they interact with one another? And how do they translate and move and shuttle from cell to cell? All of these are questions that can be answered with various biomarker discovery approaches. Now, these various approaches can also be applied across the range of early research and development, where typically we are interested in a somewhat hypothesis-free exploratory study. Maybe we are interested in associating a biomarker with a particular disease state, but then we also can consider how these approaches can be used for preclinical research through phase 1 to 3 clinical trials and even in post-approval. So as we can see in this slide here, very quickly from R&D going into the preclinical research, we're interested in identifying novel markers of disease. We are interested in how this can be used to enroll or to stratify participants within a clinical trial. And then, for example, expanding the applications of a particular therapy to other markets and other applications. So there are a wide range of technologies that can be used to perform biomarker discovery. I've bucketed a couple of them here into two main buckets, one being low throughput and a high throughput application. So on the lower throughput, ELISA and Western blots, both are typically detecting one protein at a time. The challenges with these is that if you have a wide range of samples, or a large number of proteins to detect, then this will be a very low throughput workflow. On the Western blot technique, then again, it's low throughput and also not quantitative. It's very qualitative and subjective. Now, on the other hand, mass spec has a very high throughput, can identify lots of proteins and peptides, but it does have that high upfront instrument cost. And the interpretation and the sample prep are all very challenging to perform. So with that, we would like to introduce Olink as a novel approach for protein biomarker discovery. So let's transition into that. So we would like to share here some of the advantages of Olink. So Olink is a new platform that has been commercialized by Olink and is being run as a service by Azenta Life Sciences and has very high sensitivity, enabling researchers to detect and quantify low abundant proteins. It's compatible with a wide range of sample types. Importantly, it also only requires a very small volume of a sample, typically 1 to 10 microliters. And this enables scientists to get a wide range of proteins and information from those samples compared to Western blots and ELISAs, for example, where one would typically have to pick and choose a handful of protein biomarkers to assay. And then finally is a very specific application, has a great deal of specificity due to using a dual antibody approach. And so this new application can be used for a wide range of questions that you may want to ask, whether that's understanding basic biology, involvement in a clinical trial, for example, patient stratification, identifying whether a patient might be eligible for a trial, and so forth. So unlike traditional multiplex protein-based detection approaches, where even if you have two antibodies binding, there is a great deal of potential for crosstalk between them, the Olink workflow still uses two antibodies, but these antibodies are very special in that they have a DNA barcode conjugated to them. And these two antibodies have DNA barcodes that are complementary to each other. And what this means is that when these antibodies bind to the protein target, they come within very close proximity of each other. And then these DNA barcodes anneal and then can be detected. So this enables a superior sensitivity and, importantly, fewer false positives to follow up. So here is a high-level review of the chemistry of how Olink works. I mentioned before the dual antibody binding. You see that here. When these antibodies come in close proximity, the DNA barcodes anneal with each other. And then during a PCR amplification step, we produce this PCR amplicon, which can then be detected and quantified either by next generation sequencing or by quantitative PCR. This technology has a number of flavors from the low-plex to the high-plex. So on the low-plex, these assays are called flex and target 48 or target 96. They typically are detecting between 15 to 92 proteins per assay, between 40 samples to 88 samples, and enable either an absolute quantification or a relative quantification. And each of these are available with a various customization package or off-the-shelf assays, targeting, for example, cardiovascular diseases, immune diseases, and so forth. And on the other hand, with an NGS-based readout, we're now able to massively scale up the number of proteins that can be detected in one experiment, anywhere up to 3,000 proteins by running the Explore 3072 assay, which combines a number of these panels, as you see here, into a single experiment. So we have this technology, we have the chemistry, and in order to actually implement this at the lab, we need to consider very carefully how we are going to do that and how we are going to do our liquid handling. So when we consider the requirements for success, of course, data quality is number one. We need to make sure that we have high precision and high accuracy when we are performing this experiment. We also want to minimize the reagent costs. The antibodies are expensive to produce, so we really want to minimize the amount that's needed. Plus, because this is a high-plex assay, we also need to consider that. So being able to miniaturize the reaction is very important. And then finally, we would like to perform this in a high-throughput workflow. So processing in 3D 4-well plates is really critical. And so towards this, we have been using the mosquito high volume and the mosquito low volume for tip-based transfers. Anne, in her next talk, will talk about how that technology works and the importance of the positive displacement technology. But then we are also using a tipless based approach to dispense liquids, and that's using the dragonfly, which can dispense anywhere from 200 nanoliters up to 4 mils without using tips. So here's a little bit of a deeper dive into the Olink Explore workflow. Remember, the Olink Explore workflow is the higher plex and the higher throughput workflow. So we start off with a 96 well plate. We do a 1 microliter transfer and prepare a dilution of that sample. And then we're taking 0.2 microliters of that sample and setting up an incubation with the sample and with the antibodies. So I really want to emphasize here again the importance of the accuracy and the precision of the pipetting, and just consider how challenging it would be to perform this either by hand or using a traditional liquid handler when you're trying to take 0.2 microliter. Regardless, in this step, we are using the dragonfly and the low volume mosquito, and that's what enables us to perform this low liquid transfer with great deal of accuracy. After an overnight incubation, on day 2, we then prepare a PCR reaction. We're then pooling these various plates together into a number of pools. And after following indexing and a PCR amplification step, we have our final NGS libraries. So typically this will be performed by a traditional liquid handler, but we actually very quickly saw the advantages of the Mosquito platform and quickly transitioned this from a traditional liquid handler back over to the Dragonfly, and in this case, a Mosquito high volume. And the reasons for this is that, again, we're getting more reliable data. I will show you that on the next slide. But importantly, we're also getting faster processing and reducing our plastic waste, which is very important. So here is what I was just mentioning on the previous slide about the importance of the pipetting accuracy and the precision. And we did a very simple experiment where we took a dye solution and used a traditional liquid handler to take 5 microliters of that and then ran that on a plate reader. And as you can see, there's quite a deal of variability across this 96 well plate, around 15% or so CV. Now, granted, we could have done more effort and worked on the liquid classes to optimize this, but this is what we saw out of the gates compared to what we saw on the Mosquito high volume, where we were taking a 1.5 microliter aliquot from that dye reservoir and see very tight correlation and very tight CVs across the entire plate. So we now have our chemistry, we have the liquid handling to enable this workflow. So we initially performed some feasibility testing in our lab and then moved forward with an analytical assay validation of the Olink Explore assay within our CLIA-CAP clinical laboratory. Towards this, we took a handful of samples and ran them with many, many replicates within the same plate. We also took those same samples on additional days and repeated them for an interbatch variability analysis, and also performed a concordance analysis with other approaches. So here you see the coefficient of variation of those many replicates within the same batch, typically seeing less than 10% CVs. And when we look at those same samples across multiple batches, we again see very tight CVs, in this case, typically less than 15% CV. When we compare the correlation between the Olink Explore, which is the high throughput workflow, and the Olink Target 96, which is the lower throughput workflow, we see very strong correlation, Spearman correlations of typically greater than 0.8 and even up to 0.9. And then finally, we also looked at the correlation between multiple 3D4 Explore 3D4 panels. So the Explore 3072 assay is essentially an assortment of 8 Explore 3D4 panels. And in each of these Explore 3D4 panels are a handful of protein targets that are assayed in every single panel. And this enables us to look at the consistency and the correlation of detecting, in this case, IL-6 across multiple panels. And as you can see here, the correlation between these different panels is very strong. So to conclude what I've just shown over the last couple of slides, we've deployed the mosquito and the dragonfly for Olink Explore. And as you can see, we're getting very high data quality, very high consistency. And towards this, we've been awarded a platinum service provider badge by Olink due to that high data quality and the service that we provide. But we're really interested in how we can take this further and how we can deploy the mosquito and the dragonfly for other workflows. And towards this, we've actually been using these platforms for our Olink target workflows as well. So just reminding you, the Olink target is the lower throughput workflow. And again, we are able to use the mosquito and the dragonfly to increase the data quality that we are able to produce. So I'd like to conclude my presentation with trying to pull all of this together. I mentioned earlier that when you consider the central dogma of DNA to RNA to protein, typically we're looking at each of these biomolecules in isolation. But I think it's very important to consider a sample comprehensively. And if we want to really comprehensively understand a sample and how that sample impacts an individual, then we really want to be looking at all of these levels. So we really want to be looking at DNA, RNA, and protein together. And so we showed recently at an ASHG conference that we can perform exome sequencing, methylation sequencing, single cell RNA sequencing, as well as Olink protein biomarker detection from a single blood draw. So the schematic of how we did that is shown here. We start with sample planning and then collecting whole blood. We perform a PBMC isolation and plasma isolation. The plasma is used for protein biomarker profiling using the Olink platform, as well as being stored long-term. We are also using the PBMCs to perform the exome sequencing, the methylation sequencing, single-cell RNA sequencing, and then taking all of this together in our data analysis workflows to really provide that comprehensive understanding of a sample. So to conclude today, I've shown that really, understanding a sample comprehensively is very important, and really now we are in the position to be able to perform that at the proteome. We can combine that now too with the DNA and the RNA level, of course. We have implemented the Olink workflow as a service for the mid and high-plex protein biomarker profiling. And really the use of the mosquito and the dragonfly has really enabled this workflow and enabled the high quality data that we've been able to generate. And then finally, I showed that application of bringing the proteins together, bringing the transcriptome profiling together and genomics to provide this very high-plex, very highly comprehensive profiling of a particular biospecimen. So with that, I'd like to thank you for your attention. My contact information is on the screen, although we'll have questions at the end as well. But in the meantime, I'd like to pass it over to my colleague Anne for her presentation.
So thank you, David, for this fantastic presentation and just highlighting the importance of really obtaining comprehensive data from every sample that takes you from DNA through RNA and then finally the proteome. In this part of the presentation, I want to focus a little more on the technology that underpins a lot of these workflows, why positive displacement is really truly unique in enabling some of these workflow steps and the underlying chemistries, and just highlight that the instruments used in the Olink Explore workflow are not limited to that workflow at all. These are open platforms that can provide reagent savings across a range of chemistries in the omics space. And this is definitely something to approach your service provider with if you have other chemistries that you're interested in. So the instruments the mosquito and the dragonfly discovery are often referred to as a dynamic duo, because the mosquito provides really all the unique sample transfer steps, the pipetting, whereas dragonfly discovery provides the reagent dispensing steps. And as David has outlined, this is non-contact, affording the user great savings on the consumables and just the environmental impact of their day-to-day operations. But both instruments are based on positive displacement and offer accurate and reliable low volume pipetting as well as dispensing. And in addition, positive displacement is really unique in that it's liquid class agnostic. It's ideal for handling volatile or viscous liquids, as well as your standard aqueous solutions. With that, users can access miniaturized workflows. How far users can miniaturize depends a little bit on the chemistry, but miniaturizing up to 25-fold has been realized. And of course, this very quickly translates into significant reagent savings that service providers can then also pass on to their users. The mosquito, as I mentioned, is a miniaturizing pipetting system. It comes in two flavors, HV and LV, that can pipette either from 500 nanoliters to 5 microliters or 25 to 1 nanoliters to 1.25 microliters. But unique to the mosquito is really this positive displacement tip that is highlighted on the top there. So you can see a stainless steel piston that is encased in a plastic barrel and glued onto this backing tape. And in the video below, you can see the instrument now transferring liquid from a source plate and setting down minute droplets just for visibility onto this plate lid. So you can see this is a contact dispense, and we're getting really the accuracy and precision that you would be looking for. Other things to mention is you've probably seen that the instrument can automatically index the tips to this head. We've removed a head cover there just so you can see that a little bit better. But these tips, you can use fresh ones for each sample, have zero risk of cross-contamination. The amount of liquid that is left in the tip at the end is absolutely negligible. And because we have a straight barrel geometry on that tip, it's also extremely gentle on gDNA and enzymes. There's very little shearing. So this really provides precision and accuracy for low and high viscosity liquids across the volume ranges that you really want to access with these miniaturized workflows. If we just have another quick look, there's a range of accessories that come with the mosquito. So naturally, the tips can be purchased gamma irradiated, which is ideal for omics work. We have protective covers for the spool, cooling blocks, and then for those protocols that require it, there's also a magnetic bead separation block for bead cleanup protocols. I jump ahead now. The Olink Explore protocol is fully validated for use with the mosquito and dragonfly instruments, and there are very detailed SOPs of how to utilize these instruments. But for users interested in adopting other chemistries, the mosquito software is completely open. Users can write their own protocols, but we also have a range of templates to help users get started. So you really get the best of both worlds with a range of validated methods, as well as the ability to create your own. As we've heard from David, sometimes it makes sense to transfer other chemistries onto your platform. Changing instrument now, the Dragonfly Discovery is a reagent dispenser, and you can see the instrument dispensing in that video on the top right. And you can see this is a non-contact dispense, so there's no reliance on pipette tips. This instrument could be left running all day using one set of syringes. The dispense range is from just 200 nanoliters right up to 4 milliliters. You can run both miniaturized methods as well as full volume methods. And again, it's liquid class agnostic dispensing that you're looking at. So you can do anything from buffers, glycerol, detergents, and beads, as well as your standard aqueous reagents. And the system is extremely quick. So if you synchronize 6 dispense heads, as you're seeing in the video, you can fill a plate in less than 30 seconds. The instrument is available with 3, 6, or 10 dispense heads, depending on your laboratory's needs and how much flexibility you want. You can use these heads completely independently for different reagents, or you can synchronize them if speed is more of a concern. But at any rate, you're getting really outstanding low volume dispense precision and accuracy. The dragonfly discovery, like the mosquito, comes with its own range of accessories and consumables. These are all manufactured by SPT Labtech for assured quality and supply guarantees. Everything is made from high quality polypropylene or HDPE. There's no splashing or risk of carryover. Everything is available in sterile if that's a concern. So every instrument typically uses these syringes with the plungers, and usually these open style reservoirs that are available as a standard or a loaded volume option. But for users who are interested in maybe more production type settings where they're filling lots and lots of plates, we can provide these AFR valves, which are fed via a peristaltic pump, and that then moves the reagent supply offline essentially, and you can increase the capacity to the capacity of the vessel. We also have things like cooling blocks if you're very concerned about keeping your reagents chilled during a protocol. That's also available. So we have over 40 NGS methods that our users have miniaturized. These include all the big names from Illumina, Takara, Roche, NEB, Olink kits, of course. And I just wanted to give you a flavor of some of the chemistries that are accessible. As I said, if you have any questions, contact us, speak to your service provider. We're keen to support our users to make sure they're getting the most from their instrument. But to give you a flavor, these tables, I won't go through them in detail, just highlight some of the kits, the standard volumes and the miniaturized volumes that we've been able to achieve. And these span really anything from single cell workflows on the RNA side, from low input volumes, whole genome, lots of different ones there. There are DNA sequencing methods. You'll recognize some of the big kit names here, like Illumina, Nextera, XT in the new DNA prep, the NEB Next Ultra 2 for DNA, and the FS DNA. There's Kappa Hyper Plus, and the equivalent kits, of course, on the RNA sequencing side. And then obviously not forgetting amplicon sequencing, if you're interested in that, various methods for sample and library bead cleanup and quantification, qPCR, PCR setups, and then various proteomics methods, like, of course, the Olink Explore or Agena Mass Array. So there's really a whole range of methods that our instruments can support. Please ask us if you have any questions. My contact details are on this slide below, or we have a landing page that also details this information. So with that, I'd like to thank you for your attention, and I'll pass back over to the moderator for the questions and answers. Thank you.
Great. Thank you for that interesting seminar, Anne and David. Now let's move on to the question and answer session. Just a reminder to the audience to submit your questions, and you can do so at the speech bubble icon to the left of your screen. And now the first question we have here is more directed to you, David, and that is, how could incorporating proteomics benefit genomic or transcriptomic profiling projects?
Thanks for the question. So I think I kind of touched on talking about the central dogma and, historically just performing analysis of the DNA and the RNA and putting everything together with the multi-omic approach really allows you to do a little more comprehensive profiling of a sample. And so there could be a few examples of why you might want to do that. For example, you might want to correlate the RNA expression level with the protein level. We know that they're not perfectly correlated, of course. There are cases where they are decoupled, and that's kind of important to understand, especially when considering RNA profiling is becoming more clinically relevant. We really want to understand what's happening at the protein level, but also to try and understand basic biology too, I think it could be very helpful. I think over the last couple of decades or so, a lot of genome-wide association studies have been performed to identify variants that are associated with a specific phenotype. And if we can add in protein profiling using Olink, then we can kind of actually narrow down the hits that need to be followed up. So this is called protein quantitative trait loci profiling, where you can take your GWAS hits, look to see if that protein is also dysregulated by Olink profiling, and that will significantly reduce the number of targets that you have to follow up on. There have been a number of publications around this as well. So that's kind of a flavor of, I think, why it's important, but surely there's many more applications and papers that will be published on that topic.
Okay, great. Thanks a lot for that one. I'll move on to the next question. And I think this is more one for you, Anne. And that is, how much sample or volume do you need to supply to the Olink proteomic workflow?
So, hi, thank you for the question. I think I will actually pass this over to David, because he's the one running these assays day to day. David, could you comment?
Sure.
Please?
Sure. Yeah. So that really depends on the workflow that is being run. Typically, we need 1 microliter to run the Olink target workflow, and around 10 microliters or so for the Explorer. We would typically ask for more just to have more on hand, but it's a very small volume needed.
Great, thanks a lot for that one. And another question about your experience with the Olink workflow, maybe David, and that is, what is or was your biggest challenge with setting up the Olink workflow in your lab, and how did you address this practically or technically?
Yeah. So it's a pretty novel workflow, and it's a pretty complex workflow. I think I kind of gave a pretty high-level overview of it, but when you're at the bench, you have lots and lots of 3D 4-well plates, a lot of liquid transfers taking place, and so we were, on the one hand, really grateful to have a really good relationship with SPT and with Olink to work on the validation of those liquid handling scripts together. That was quite a lot of work to test those methods, but went pretty seamlessly by working together. But then, coming back to the number of plates I just mentioned, then that can also be a challenge in the lab, and keeping track of those plates. So one of the ways that we addressed that was incorporating barcode tracking of our plates and incorporating that to our LIMS system, and that's really helped with the processing of this workflow.
Okay, great. Thanks a lot for that. And I'll open the next question for either of you, and that is it possible to customize Olink panels?
Yes, I probably can take that one. It is possible. So there's 2 different approaches that one can take. One can design a pretty big panel, maybe around 40 targets or so. That is one thing that we can work on together with Olink. The alternative is that we can make a smaller panel. It's called a flex panel, where the researcher can select from, I think is around 200 pre-validated proteins, selecting around 20 or so of those to put into a custom panel. So those are the current options that are available.
Great, thanks a lot for that one. And the next question I've got here is, do either of you have any tips and tricks for sample preparation, storage, and control?
So I guess I'll maybe talk about that as it relates to the Olink workflow in particular. And so we are a service provider, right? We have a team of PhD scientists that will help the researcher, though, with the experimental design of their study. And that really goes into every level of detail, thinking about the number of samples that are needed, thinking about the sample prep, which is pretty important. A wide range of sample types can be used for this workflow. Typically that is plasma or serum, which is pretty straightforward to process, although we do have some guidelines we can pass along. But we can also use this for cell culture, supernatants and tissue lysates and so forth. And that's where it gets a little bit more tricky. So again, we have some guidelines that we can pass along, some written guidelines, but some of the main things to consider there will be the controls that are being added. And so, for example, going back to the cell culture medium and using that as an input, then it's really important to understand the nature of the medium being used, because you don't want to be detecting the growth factors that you're adding into your cell culture. You really want to look at what the cells themselves are producing. So those are some of the challenges that are important to be aware of ahead of time, and that's going to, again, how we can help with the experimental design.
Great, thanks a lot for that insight there. And I've got another question directed for you, David, and that is, why did you sometimes use mosquito HV and sometimes mosquito LV?
Yeah, so these are, I would say fundamentally the same machine. I think maybe Anne maybe has a slightly different opinion, but I think is kind of, specialized for the liquid volume, right? So they're both able to take the same type of plates. They're both able to take the same number of plates, but the high volume is really geared towards on the higher end of liquid transfers, between 0.5 to 5 microliter, and the low volume is really designed for the really low volume 0.2 microliter type range. And so we've picked these two machines and implemented them into those two different steps of our workflow, where we're using the low volume instrument for the day one on the incubation setups, because those volumes are so low that needs to be transferred. Whereas on day two, the volumes are a little higher and more amenable to the mosquito HV. And then the other nice advantage of that is that we're still able to have some separation between our pre and post PCR to have a unidirectional workflow. So again, that kind of helps with the overall data quality and the robustness of the assay.
Yes, and maybe if I can add to that, that was actually a really great answer. It's really all about choosing the right tool for the job and for the chemistry that the user is after. There's clearly a little bit of overlap in the volume range between these two instruments. So sometimes it can be a case of using one or the other. Sometimes we might recommend that one would be more suitable than the other one. But I think that's typically really workflow and context specific. And as an instrument vendor, we would work with the user to figure out what would be the best solution for their specific goals.
Great. Okay, I'm afraid I'm going to have to wrap it up there. But thank you again to Anne and David for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope that you've all found this a worthwhile session. And if you have any other questions, please feel free to email me at editor@selectscience.net, and I'll follow up with your questions for our speakers. And remember, you can also download a certificate of attendance in the Related Resources tab to the left of your screen. If you would like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days. Goodbye, and thank you once again for joining us.